Thank you. It's my pleasure and privilege to be part of the International Cancer Conference 2025 today. I'm Dr. Saumya Pandey, PhD Life Science. Recently worked as Head Department of Clinical Research, IndiraIVF Hospitall, Udaipur, Lucknow, India, and it gives me immense pleasure to be here today to present my talk titled Human Papillomavirus Prevalence and Genotype Distribution Among Cervical Cancer Susceptible Women of North Indian Ethnicity in Gardasil Vaccination Era, Public Health Impact, and the emphasis would be on the scientific sessions, prevention, epidemiology. May I request the slide transition? Next slide, please. An overview, cancer research snapshot. Human papillomavirus is the major etiological agent of cervical cancer, a leading cause of morbidity and mortality in women worldwide. Screening strategies for reducing the burden of HPV-mediated carcinogenesis are emerging as effective means for cervical cancer control and prevention in developing countries. And I have publications as the first author in gynecologic oncology with emphasis on HPV-mediated cervical cancer, International Journal of Gynecologic Cancer 2010, Molecular Biology Reports, and Asian Pacific Journal of Cancer Prevention. So there is an urgent public health challenge worldwide to cost-effectively manage the disproportionate share of morbidity and mortality associated with human papillomavirus-mediated cervical cancer in susceptible, vulnerable population pools of women of varying genetic landscapes and lifestyles. Cervical cancer control and prevention strategies are being adopted globally to reduce the disproportionate share of HPV-mediated cervical cancer in the Gardasil vaccination era. Another publication with emphasis on HPV cervical cancer and Gardasil vaccination, Public Health Impact, by me as the senior lead author in Journal of Community Health. Next slide, please. The complexities associated with human papillomavirus is indeed integrating with the 2D structure of human papillomavirus, the 3D structure, with lots of complications going at the regulation of virus gene expression and viral replication, disruption of cell growth cycle, aberrant cell proliferation rates, the interplay of p53 telomerase, retinoblastoma, the capsid proteins, the viral integration, DNA replication, the viral and host synergistic or aberrant interactions at the transmembrane level, frequently disrupted site in case of integration into the host genome, upregulation and downregulation of gene expressions, inflammatory cytokines and chemokines being overexpressed, an array of receptors, primarily toll-like receptors at the transmembrane level, recognizing the human papillomavirus oncoproteins, assembly and release, and the membrane signaling proteins with the complexity associated with viral replication, integration, and subsequent initiation of cancer, and aberrant cell proliferation, cell death, autophagy, leading to eventual cervical cancer in susceptible cohorts of women of varying genetic landscapes. And I have reviewed an elegant paper focusing on HPV and cervical cancer published in Frontiers in Oncology. So there is an urgent need for all the delegates today at ICC 2025 to join hands for the fight against HPV-mediated cervical cancer, as well as other cancers that are emerging as the primary risk factors affecting susceptible cohorts of varying genetic landscapes and lifestyles. Next slide, please. Cervical cancer is a leading cause of morbidity and mortality in women worldwide, with the high-risk HPV types being 16 and 18 as the major etiological agents of cervical cancer. And there are approximately 130,000 new cases of cervical cancer in Asia Pacific, my home country, India, per year. And the age standardized incidence rate is approximately 30.7 per 100,000. So a relatively high rate, and the link between genital HPV infections and cervical cancer was first demonstrated in early 80s by Harald Zerhosen, a German virologist, and HPV was considered as a major causative agent of cervical cancer. And there has been a lot of thrust on development of prognostic and predictive biomarkers in cervical cancer management and the emergence of vaccines, primarily the quadrivalent and monovalent vaccines, and Gardasil vaccination era has indeed led to a significant reduction in the increasing disproportionate trends of cervical cancer morbidity and mortality. Next slide, please. I have reviewed cervical cancer and HPV model in Frontiers in Oncology, where we see the oncoproteins HPV 16, 17, and 18 as the major oncoproteins, with E1, E2, E3, E5, E6, E7, and the L1, L2 code proteins, with Cervarix and Gardasil being the two primary vaccines for effective management of HPV in girls as well as women of reproductive age. The protein-based and peptide-based vaccines, the nucleic acid vaccines, whole cell-based vaccine, and the live vector vaccine are indeed protective for cervical cancer management. Next slide, please. My innovative evidence-based exploratory study had the primary objective to identify HPV prevalence and genotype distribution among cervical cancer susceptible women, along with Gardasil vaccination awareness for cervical cancer prevention in Asian Indian women during random population screening in genetically susceptible cohorts of North Indian ethnicity. Next slide, please. Methodology: cervical vaginal exfoliated cells and pap smear specimens were collected from 890 women of North Indian ethnicity residing in Lucknow, Uttar Pradesh, and adjoining areas during random population screening. HPV viral growths in clinical specimens were determined by the hybrid capture HC2 HPV DNA assay, which is approved by FDA, and subsequently positive negative borderline HPV status was determined with minimal selection bias and considerable specificity and sensitivity. So further, a pilot structured questionnaire-based survey was conducted amongst a stratified subset of HPV positive women in my innovative clinical research study, with a statistically powered sample size of 819 women, with bilingual interviews in either Hindi, which is the local dialect, or English, the universal mode of communication. Subsequently, the awareness of HPV-mediated cervical cancer and the knowledge of Gardasil vaccination was assessed in terms of yes, no, and no response. So the structured bilingual questionnaire was indeed instrumental in eliciting the true response rates amongst susceptible women. Next slide, please. Immunobiology of toll-like receptors. This is my first paper, published in Immunology and Cell Biology on August 6, 2006 in Biomedical Sciences and Life Sciences from Creighton University School of Medicine, Nebraska, United States. And I was just discussing that we may therapeutically target the enigmatic array of toll-like receptors, which are a family of evolutionary conserved pathogen recognition receptors at the transmembrane level. They are the primary sensors of invading pathogens, such as human papillomavirus, the different oncoproteins, the primary ones being HPV 16 and 18, and TLRs identify molecular motifs of infectious agents, the pathogen-associated molecular patterns. They have the MyD88 and the TRIF-mediated cascades, leading to inflammatory responses in the aberrant physiological milieu, cancerous, precancerous to full-blown cancerous cell in the host. So we may therapeutically target toll-like receptors in conjunction with cell death pathways, primarily apoptosis and or autophagy, in the next few years to cost-effectively design and develop novel, patient-friendly, evidence-based immunotherapeutic modalities for effective cervical cancer management in women of varying genetic landscapes and lifestyles. Next slide, please. Autophagy in cancer pathophysiology. Another cell signaling pathway that we may target with the enigmatic array of toll-like receptors is the autophagy machinery, with cellular stress in the cancerous, precancerous trait, with the physiological milieu of the normal cell being tilted from hyperproliferation to senescent to a cell death mode. So cellular stress leads to the overexpression or underexpression of key proteins in apoptosis cell death pathway, primarily BCL2. And in autophagy, there is a complex interplay of PI3 kinase and the Beclin-3, which is the major central regulator of autophagy, with proteins ATG1, 2, 5, and 7, the microtubule-associated light chain 3, LC3, 2, and 1, leading to the autophagic flux, the tilt in the cell fate from cell proliferation to cell death, and eventually a full-blown cervical carcinoma, with the patient being even, unfortunately, having an early demise or death. Microtubule-associated protein light chain 3, ATG5 and 7, and Beclin-1 studies have also been successfully published by me as the senior lead author in the recent years in colorectal carcinomas. So lots of cancer research going on these days with thrust on the immunotherapeutic targeting of metabolic, biochemical, cell signaling cascades, primarily the toll-like receptors, autophagy, not only in HPV-mediated cervical cancer, but also in lung cancer, in hepatocellular carcinoma, as well as colorectal carcinogenesis. So we all need to join hands today at ICC 2025. And once again, a big thank you to the Magnus Group for providing an excellent, dynamically collaborative platform for the exchange of innovative, emerging, and evolving ideas in cost-effective cancer care. Now, autophagy, the previous slide, please. Maybe go on to the previous slide. The slide before, these are taking the slides forward. I need to go to the earlier slide. May I request the technical support to May I request my co-host to upload the presentation again? It was midway and I was just discussing the slides. We need to discuss the results and the takeaways. Yes, the further slides. The one after this. We already discussed autophagy. I appreciate. Yes, this is the one. Now we need to go ahead with this slide. Yes. So autophagy in HPV-mediated cervical cancer pathophysiology is a process of cytoplasm and cellular organelle degradation in lysosomes, with the cell fate tilting towards cell death. And we may design predictive and prognostic biomarkers with the interplay of toll-like receptors, autophagy, and apoptosis for effective cancer care and treatment. Next slide, please. Results of my innovative clinical cancer research study, HPV prevalence in Asian Indian women of North Indian ethnicity was 11.7%. HPV genotype distribution was accurately evaluated. 751 out of a total of 890 women, 84.4%, participated in the HPV screening program, were HPV negative. 104, 11.7% tested positive, while 3.9%, a total of 35 out of 890, showed borderline infection status. Furthermore, in the HPV positive subsets, 104 women, 18, that is 17.3%, displayed strong positivity trends. HPV positivity tends to increase with age in North Indian women. Higher the viral load with increasing age, higher is the susceptibility to HPV-mediated cervical cancer. So overall, the response of participants was well defined using the conventional innovatively designed bilingual questionnaire in Hindi and English with HPV-mediated cervical cancer awareness in terms of yes, no, and no response, amongst the study subjects being 43.7%, 44.7%, and 11.6% respectively. Furthermore, in response to knowledge awareness of HPV vaccine Gardasil, out of 103 subjects, 28% answered yes, while 37% and 34% stated no and no response respectively. So we need to push forward the HPV vaccine Gardasil related awareness, and the awareness campaigns need to be fostered in a collaborative fashion for fighting HPV-mediated cervical cancer in the years to come. Next slide, please. HPV infection status in North Indian women, the negative, positive, and borderline status. 751 women were HPV negative, 104 tested positive, and 35, that was 3.9%, showed borderline infection status. At higher the viral load, higher is the susceptibility to cervical carcinogenesis. Next slide, please. HPV positivity in North Indian women, 104 study subjects with 86 women testing HPV positive, while 17.3% showing strong positivity status. Next slide, please. Schematic depiction of strong HPV positivity in a total of 18 out of 890 women as indicated by the HPV cutoff ratios. These are the cutoff ratios, and 500, 1,000, 1,500, 2,000, 2,500, 3,000, and 3,500, reaching the maximal between 3,000 to 3,500. So high HPV cutoff ratios depicting the strong HPV positivity trends. Next slide, please. My promising HPV viral load genotyping may help in identifying women at risk of developing cervical cancer. However, cost-effective HPV screening protocols with a wider population coverage are warranted so as to reduce the burden of cervical cancer in women worldwide in the Gardasil vaccination era. Therefore, understanding the ecological diversity of human papillomavirus, prevalence and genotype distribution among genetically susceptible populations in different geographical regions worldwide is essential for optimizing HPV screening, vaccination, and maximizing the cost-effective, public health-oriented strategic efforts for pragmatic, evidence-based cervical cancer prevention, including the future design of novel immunotherapeutic evidence-based regimens, tailor-made precision oncology. We may also couple the novel evolving artificial intelligence algorithms with cervical cancer precision-based oncology with predictive and prognostic biomarker studies, causal association studies for effective HPV-mediated cervical cancer management in women of varying genetic landscapes and lifestyles globally. Next slide, please. Further biospecimens. HPV-mediated cervical cancer patients' blood tissue, DNA, and oocytes may be collected and frozen stored for long-term usage in individualized pregnancy, family planning, post-cancer chemotherapy treatment, and/or conducting multicentric public health, gynecologic oncology, causal association prevention epidemiology studies in fertility preservation among cervical cancer patients, susceptible women of varying genetic landscapes and sociocultural exposures. So there is a lot of thrust on even addressing the psychosocial, psychosexual, and the distress associated with HPV-mediated early versus advanced stage cervical cancer in women of reproductive age, childbearing age at many countries with fertility preservation issues that may affect in fulfilling the cherished dream of having a complete family, of having a child with the cancer patient. So biobanks and biospecimens need to be stored in biobanks, and biorepositories may be helpful in fertility preservation and the underlying psychological mental health issues associated with HPV-mediated cervical cancer and fertility preservation may be addressed by developing biobanks or biorepositories for cost-effective oncofertility management in women of childbearing age of varying genetic backgrounds, landscapes, and sociocultural exposures. Next slide, please. Impactful publications by me as the senior lead author. Fertility, sterility, toll-like receptors and tissue inhibitor of matrix metalloproteinase genetic variants as predictors of cervical cancer and fertility. Evaluation of toll-like receptors and TLRs 3 and 9 in cervical cancer susceptibility, psychooxygenase 2 gene polymorphisms, and risk of cervical cancer in North Indian population, gynecologic oncology, therapeutic targeting of TLRs 2 and 4, the gene polymorphisms for cervical cancer susceptibility in North Indian women, offering fascinating avenues for cost-effective predictive biomarker development and effective patient-friendly cancer care in vulnerable cohorts, and fertility, sterility, tobacco, and environmental carcinogen as a significant predictor of infertility, with lots of public health perspective in oncofertility in Indian as well as the global scenario. Next slide, please. The psychology and mental health care issues need to be addressed. What I was just discussing, fertility preservation in HPV-mediated cervical as well as breast cancer patients of varying genetic landscapes and sociocultural exposures, for psychosexual issues to be addressed effectively by developing biorepositories and creating biobanks for fertility preservation. Next slide, please. May I request a quick transition to the next slide, please? Acknowledgements. The previous slide, acknowledging National Institutes of Health, United States of America, Creighton University School of Medicine, Nebraska University of Texas Medical Branch, and New York Presbyterian Weill Cornell Medical College, along with the Magnus Group and ICC 2025 for providing an excellent platform today for presenting my innovative research on HPV-mediated cervical cancer in the Gardasil era. Thank you.